Thank you, Mr. Chairman. Um, I've already started talking about the aortic root and why it is important that it is the living characteristics are important. I will try and be brief because there are two very, very interesting papers coming up by Navaneeth and Rio Tori. So the aortic valve performs extremely sophisticated functions, and the important thing we're realizing more and more is that this is dependent on its specific geometry, but also very importantly on its living components. And I'll try and show you why that the case, and why is it important, because it affects myocardial function, coronary flow, and blood flow distribution, and it translates into clinically important clinical endpoints. And these we have heard already from Professor Brand and from Dr. Abdulmullah that it is a valve mechanism. It's not just a leaflet. And that all these component parts of the valve actually change their size and shape during the cardiac cycle, and why some time ago, and I described it as a tale of dynamism and crosstalk. They actually talk to each other and they change their shape. And the geometry, and what is dynamism? Dynamism could be passive or active. The passive one is due to geometry, but the active one is due to living characteristics. And it translates into this beautiful flow characteristics. You will see a lot more from Rio Tori about flow. But this is an MRI picture showing the sophisticated function of the flow, and also the flow, if you measure it and look at it by MRI, as Kilner did at the Brompton, you see that it's not only the outflow, but also the inflow, which is beautifully coordinated to produce this type of smooth flow. The valve changes its size and shape in different parts of the cardiac cycle, and this is a study from Stanford showing just that. This is a study from our lab by Ismail al-Hamamsi showing, interestingly, that the valve actually changes its mechanical properties, its stiffness, in response to secretum, to mediators from the circulation like nitric oxide or endothelin. And is that important? We think it is, if there is a lesson from nature, because the swift moves the wings in every direction, but importantly, as is said here in this seminar paper, the swift can change the shape of the wings during flight, and that is what is meant by morphing gives the swift exquisite flight control. And by doing that, not only the position and the shape, but also the size of the wing is constantly changing. And the result is that the swift can have this exquisite control over its flight path, way beyond anything you think. And that is due to the change in shape and size. And does the aortic valve do the same? Maybe it's a big jump, but we do have some evidence, and this is work from Adrian Chester in our lab, showing that the aortic valve cusps actually do respond in a dose-dependent manner to a variety of mediators present in the circulation, and that the valve cusps have nerves which are active and have a variety of mediators, like PGP is the total, the number of neuronal fibers, but sympathetic, MPY, tyrosine hydroxylase, sensory, and parasympathetic. Importantly, the movement of the aortic root, as this work by Davis and Kim Parker from Imperial showed that the main movement of the aortic root affects coronary flow in a big way. I recommend this paper for you, to you from Davis and Kim Parker, which was published 3 or 4 years ago, and that's from the same paper. How can these valves move and do all these things? If you study the molecular aspects of valve interstitial cells, like this was done in our lab, you find that there are many molecules which govern both orientation but also can affect movements, like you see here. That is, whoops, I lost everything. Like you see here, these are smooth muscle alpha actin in the valve, which is capable of moving. It's not only smooth muscle alpha actin, which is a contractile protein, but actually we have shown that in the valve interstitial cells there are contractile proteins of striated muscle, which is typical for the valve and not present either in myocardial cells or in skeletal muscle, which is fantastic. And if you have valve interstitial cells and you look at them, stimulate them with something like endothelin-1, you can see that they start to show calcium fluxes. We know that the frequency of this release of calcium translates into several things. 1, contraction, B, gene expression. So these cells are very, very active indeed. They're not only active in their own right, but they communicate with each other by this very intricate system of communication. This from valve cusps showing the interstitial cells, and the molecules responsible for this communication was studied by Najma Latif in our lab, but time will not allow me to go into that in great details. It's not only the valve valve cells which are active, but also the ECM, the extracellular matrix, which have a very specific composition and actually communicates with the valve cells, as you will hear from Navaneet, that the actual extracellular matrix can control the behavior of the cells. How does that translate into clinically relevant findings? Here we have actually compared the only valve which guarantees continued viability of the valve, which is the Ross operation, and we did in a randomized finding, and that was published in the Lancet, showing that it actually, oops, I don't know why, that on the cover of the Lancet they put our findings support the hypothesis that a living valve implanted in the aortic position can significantly improve long-term outcome in adults with aortic valve. So the editor of the Lancet thought that this was relevant enough to put it on the cover of the Lancet. And that was because we have shown that the only valve which can guarantee long-term survival similar to the rest of the population was the living valve, and that this also improved quality of life. So I don't want to be long because I want to allow time for my colleagues. In conclusion, valve function is extremely sophisticated and involves very complex biological systems, and that recreating these functions can translate into significant clinical benefit. And although significant progress has been made, and actually next week in Mykonos we're having a whole meeting on the biology and tissue engineering of heart valves, and you are all cordially invited to come to Mykonos to listen to all these papers on both morphogenesis as well as the biological function of the valve. But we really do need further concerted efforts for further research. Thank you very much.
The relation between the hemodynamics and the geometry of the heart, and also it was found that not only the valve shape is important, but also the shape of the aorta. For example, the bulging of the sinus valva is important in order so that the cusps do not stick to the wall when they open, and there is vortices behind the cusps, between the cusp and the sinus valva, which helps the closure of the valve during diastole. So these correlations are really amazing, and shows how the creator is really we really should.
Absolutely, I mean, you're absolutely right. The work of Bellhouse and Bellhouse from Oxford showed that the vortices stabilize the position of the cusp because they supply closing power, if you like, to the cusps, and indeed the cusps begin closing during ejection when there is deceleration, and therefore at the end they just have a very small distance to go. And similarly, what you've said, that recreating the sinuses in the valve preserving operation is vital, and the dynamism is very important, even if it is passive dynamism.
I apologize for a question that may be difficult to answer briefly. What are your thoughts regarding why valves calcify? Active process, passive process, or combination?
That's an excellent question. There is evidence that valve calcification is indeed an active process, and it's genetically controlled by a program which I've started to talk about with the RUN-X. In the journal, which is outside here, the first article is by Adrian Chester, talking about the molecular mechanisms of valve calcification, and it talks about the importance of the valve interstitial cells in the program of calcification, as well as invading cells. But now we're realizing also that the extracellular matrix is extremely important, and work by Craig Simmons shows, for example, that differentiation of cells, be it just progenitor cells from anywhere, depends very heavily on the physical properties of the scaffold and the matrix, and you will hear from Navaneet about scaffolds in tissue engineering, because if you put the same pluripotent cells on a matrix which is very soft, it differentiates into a brain cell. If you put it on a medium stiffness, it produces cartilage. If you put it on a stiff matrix, it produces an osteoblast. So this constant talk between extracellular matrix and cellular components is essential, and it is indeed an active process. It was oversimplified saying it's very similar to atherosclerosis, and if you give statins, it will stop it. Randomized trials showed that mixed. It's a lot more complex than that, and there are. It is an active process. The answer to your question, I think.
Further questions? I have one question with regard to the innervation. I mean, how important is innervation for the valve? I mean, given that a transplanted heart lacks this innervation, right?
Sorry?
I mean, a transplanted heart would lack this innervation, correct?
Not correct, sorry. Interesting, sorry, sir. Because you're absolutely right, the transplanted hearts in the valves, unlike valve allografts, do not degenerate. And the reason for that is that we think is that with immunosuppression, all the valve components continue to be alive, and therefore all the biological functions I've talked about continue. Now, including the nerves are not entirely exogenous, because there are a lot of local ganglia which supplies sort of nerves which are not dependent on exogenous innervation. And some studies we have done and others show that hearts do re-innervate anyway. So after a period of time, nerves from outside do invade the heart, but even if they don't, the local nervous mechanism consists. It has not been looked at, so what I'm saying, it is a bit speculative.
Thank you, Prof, for this very enlightening and future aortic valve concepts. My question is about translational research in all this, trying to put these fundamental understandings into benefits of patients. Are we very far in that direction?
No, I think that this justifies the use of valves which are living, like the Ross operation, and why the Ross operation, you have patients living 35 years later, very happy, more active, and it needs more and more research, and it justifies why we put a lot of efforts in tissue engineering, in creating a living valve by tissue engineering, and you will hear the next talk about tissue engineering. So that's translational.
So that's an invitation for the next talk, I think.
Yeah, it is.
Yeah.